Okay, guys, we're still busy with this exam here. We've done from questions 1 through question 16. This is where we stopped. Just to remind you, this exam paper was written in 2025, May, June, okay? May, June 2025. It is paper 1, multiple choice, Cambridge International AS and A level. Right, so today we want to proceed. We want to continue with this exam here. We are now doing 17. 17 reads, which of these substances can pass directly through cell surface membranes without using a carrier protein or channel protein? So you know that channel proteins, proteins are charged, okay? They are polar, right? Their net charge, okay? Okay, they are polar substances, all right? They are polar, so they allow ions to pass through them, okay? So we have got options here. We've got 1, 2, and 3. 1 is these two ions here, potassium ion, okay, potassium ion here, and chloride ion here, okay? So you know that these ions here are polar, right? These are polar, okay? Given that they're actually coming from ionic substances, okay? We are talking about salts here, okay? So they dissociate into ions. This is a cation and this is an anion. They've got the charge, so they are polar, all right? So yes, it needs to be carried through a carrier protein or channel protein. So number one. So the question here is which of these substances can pass directly through cell surface membrane without using carrier protein or a channel protein? See, number 1 cannot pass through because the cell membrane is made up of two. It's made up of phospholipids, okay? There are 2 phospholipid layers. We call it a bilayer because of that, okay? A bilayer, so 2 layers of phospholipids. So now the name phospholipids conveys a specific meaning, okay? So this is how it looks like, okay? The cell membrane will look kind of like this. One phospholipid here and another layer here. So this region here is hydrophobic. It's made up of fatty acids. So 2 regions that are hydrophobic. So this is the cell surface membrane, and this is the phosphate group. So molecules cannot pass through this region because this region is non-polar because it's made up of lipids, okay? It's made up of lipids, and lipids are non-polar substances, all right? So 1 is not true because of that. So 2 is apolar. CO2 is carbon dioxide is apolar. It's the only one that can pass through. Regarding 2 and 1 and 2 in this case, only CO2 so far can pass through. Let's look at number 3. So number 3 is this is glucose, right? Glucose is also a polar molecule. So the answer can only be 2. So the answer is 2 here. So what you have to understand is that channel proteins are there to make sure that substances that cannot pass through, okay, straight away or directly through the cell surface membrane can do so by means of channel proteins or carrier proteins. So the answer is going to be 2, which is D. Do you understand? So let's proceed. Now, this was 17. Now, we want to do 18. 18 reads, which of these statements about facilitated diffusion are correct? Number one, it is limited by the number of transport proteins. So facilitated diffusions are for substances that cannot pass directly through the cell surface membrane, so they need to be aided, they need help. Okay, they need an aid, a help. So channel proteins do that, right? So if you increase the amount of transport proteins, you can also increase the amount of you can also increase, in this case, the rate of facilitated diffusion, because they're saying which of these things about facilitated diffusion are correct. It is limited by the number of transport proteins. So if you increase the amount of transport proteins, you are going to increase the rate of diffusion here. So most substances can pass through, okay, the transport protein. So the answer is yes, it is correct. 2, it transports molecules against their concentration gradient. No, facilitated diffusion happens down a concentration gradient, right? It is down a concentration gradient, which means from higher concentration to lower concentration. So the answer is no. And then number 3, it requires a source of ATP. And the answer again is no. Facilitated diffusion, as I told you earlier, happens down a concentration gradient, right? So only molecules that move up a concentration gradient, which is against a concentration gradient, requires a source of energy, requires a source of ATP. Okay, a substance that must move from a lower concentration to a higher concentration is not a spontaneous process. It is up a concentration gradient. So from lower to higher requires energy, so requires ATP. All right, so the answer here is only 1. It's only yes is for 1. Sorry guys, it's only 1, and I actually wrote yes here. So it's only 1. Okay, 1 is the only one that is correct. Therefore the answer is C, 1 only. All right, and then we also have a cell absorbs amino acids. The cell then synthesizes and exports a digestive enzyme. Different cell structures are involved with different stages of this process. Which row shows a possible sequence of cell structures that amino acids pass through? So it's gonna be, okay, so it says a cell absorbs amino acids. So it absorbs amino acids. These then are used to synthesize a protein, and this protein that's going to be synthesized is going to be a digestive enzyme. So which cell structures are involved with different stages of this process? So the first one is going to be the rough endoplasmic reticulum, right? So then after that synthesis, it's going to be taken out from the rough endoplasmic reticulum to the Golgi body. So second, so it is processed. Sometimes they are modified there in the Golgi body. Then after that, they can form their vesicles. Secretory vesicles is number 3, and then eventually they go to the cell surface membrane. They fuse with the cell surface membrane, and then the protein is gonna be used somewhere else. Okay, so it's going to, so that's why we're calling it. That's why it says there that the cell then synthesizes and exports. So it is made in the cell and then taken to another location. Okay, so it's going to be produced by the cell. So it starts in the rough endoplasmic reticulum. From the rough endoplasmic reticulum, it is modified. Okay, the final process is actually done in the Golgi body, where the modification takes place, and then there is going to barred off from the Golgi body as vesicles, and then it fuses with the cell membrane, and then it goes out of the cell like that. All right, so the answer is C. Okay, that's it, guys. Today we are going to stop here. Thank you so much.